Hello everyone. Today in this session, let us see how we can simplify column chromatography along with its principle and instrumentation. Let us see how are we going to carry out column chromatography. Here is the column in which we are going to perform column chromatographic technique. The column may be of glass or plastic or stainless steel. So we are using glass column generally because we can see through it. Okay, the first thing what we are going to do is place a cotton wool or glass wool or an asbestos pad at the bottom of the column before packing the stationery face. And why we are packing this asbestos or glass wool? The reason is it will prevent the stationary phase being washed out of the column. So next step is pack the column with the stationary phase. The packing can be done by two methods, dry packing or wet packing. In wet packing, we pour a slurry of solvent and adsorbent meaning stationary phase to it. Next is dry packing. In that, fill the column with the dry adsorbent. After packing, the adsorbent should be wet in the column to prevent the cracking of the column. Okay. So, generally we use silica, alumina, cellulose, etc. as stationary phase. After filling, place a filter paper on top of this uh, filled column. Okay. Next is the introduction of the sample. So before filling the sample, remove the excess solvent from this column. Okay. So the excess solvent is removed from the column and the mixture to be separated is dissolved in minimum amount of appropriate solvent and add carefully on top of this adsorbent. So this is our sample to be separated, and to develop the column, add the mobile phase or solvent on top of this column. This is a mobile phase. So solvent or mobile phase may be single one or a combination of different solvents. Here we place our sample which is dark green colored. Suppose it has three components. Okay. So the principle behind the separation is affinity of each component for the adsorbent and for the solvent. The component which has more affinity towards the stationary phase stay longer in the column, and the component which has lesser affinity towards the stationary phase very quickly separated and pass through the column and get separated fast. So here you can see one component gets separated which is blue in color, which has very less affinity towards the stationary phase but has more affinity towards the mobile phase. So here you can see this blue component which is having very less affinity toward the stationary phase, and see this is blue component has traveled after some time to this point, and after some time we can collect this separated component in a beaker. Before that you can see another component getting separated which is violet in color. So this compared to this blue band, this is having little more affinity toward the stationary phase. Okay. So next we are going to collect the first component which is separated, and you can see the other component is also almost ready to get separated. So once we separate this component, the next component we can separate, and meanwhile you can see there is a third component or the last component is getting separated which is green in color, and at last you can separate that green component also. So out of these three components, blue has very lesser affinity towards the stationary phase. That is why it gets separated faster, and this third green component which is having more affinity towards the stationary phase and less affinity towards the mobile phase. So mobile phase took more time to carry this from the uh, top of the column till here. Okay, so this is how we separated our three components, and each separated components we can collect in separate petri dish or beaker depending on the sample. So this is the simple illustration of packing and instrumentation of column chromatography and the working of this. Thank you.